My name is Emie Roy, and I'm joining the session from Sydney, Australia. And it's amazing to see that we are about to hear a talk from Dr. Robert Barigye, who has actually worked across so many different countries in the world. And he's actually done quite a lot of research in, a lot of immunological areas. So really looking forward to hearing from him. And thank you so much for having me on board.
Thank you very much.
Thank you, madam.
Go ahead. Go ahead.
Sorry?
Hello.
Can I go ahead?
One second, sir. Babji, sir, are you there?
Yes, I'm here.
I'm handing over the session to chair and co-chair. Sir, please.
Okay. Thank you very much. At this time, as has been said, we have a session on proposal to pursue One Health strategy in the study and management of animal Coxiellosis or Q fever in the United Arab Emirates. Now we have a speaker, Dr. Robert Barigye. He shall be presenting the lecture right now. Session is handed over to Dr. Robert Barigye.
Thank you very much. I'll go ahead and share my content, but I'm very happy and privileged to be here with you guys. And I extend my thanks to Dr. Maya, who invited me. It was at short notice, but I hope that what I share today will be of some use to all of us. So let me go ahead and share my content. Can you all see my content?
Yeah.
Okay.
Yeah, sir.
Thanks for the kind words, all those who have introduced me. Obviously, now everyone knows I work here. I'm assistant professor at the Department of Veterinary Medicine here within the College of Food and Agriculture. I've been here for 6 years, having arrived from Northern Australia, where I had stayed for 4 years. And before that, of course, I had stayed 8 years in the US, doing the different things that were talked about. So my presentation today, this work was instigated by my quest to do research when I arrived here. When you arrive in the UAE, you realize actually there is very little livestock research going on. And what I thought was actually to get some baseline data before I jump into any kind of research. And I'll be sharing with you that work towards the end of the presentation. The first part of the presentation to appraise all of us about what Q fever or Coxiellosis is. I'll be talking about the etiology of the organism itself, the history of Q fever, and the epidemiology of the disease, which is rather complex, the ways in which the disease is transmitted, the disease in humans, in animals, different species of animals, and then the prevention and control. Then in the second part is where we'll talk about what actually I've been doing here. Of course, initially I did a thorough literature review and found actually there was a lot of Coxiellosis, at least reported in animals, and a few cases reported in humans in different countries within the Middle East. And then I'll be talking about the cell surveillance studies we've done in cattle, sheep, and goats. And then the next proposal that we are supposed to be embarking on using this One Health strategy to approach the research and later on, of course, the management of the disease in the country. So I'll go ahead. Q fever is caused by a gram-negative bacteria. It's an obligate intracellular pathogen. As you can see in the picture here, is Coccobacter in morphology, and it belongs to the family Coxiellosis, subdivision Proteobacteria. The first scientists who worked with this organism actually had classified it as a rickettsia, and later on realized this was a different organism altogether. One unique characteristic of the organism is it's very stable in the environment, which plays into the epidemiology of the disease. In some cases, it can stay in the environment for months and months, especially like in urine, dried feces, and so forth. And then the organism has two stages in its life cycle. the small cell variant, which is a spore-like structure, which would explain why it's so resistant in the environment. And then large, which is found intracellularly within the infected host. About the history of this disease, actually the disease was first described in Australia when a gentleman called Derek, working in Queensland, actually saw a flu-like disease in a cluster of urban workers. And he investigated and actually isolated the organism. I mean, I admire his case way back in 1935 to how quickly actually cultured this organism, which is so deadly. And around the same time, another group of workers, Davis and Cox, working at a USDA laboratory in Montana, also described the same organism. And when they compared the notes, the group in Australia and the one in the US realized actually they were working on the same organism. Since then, Coxiella barnetti, the cause of kill fever, has been reported almost in every country with the exception of New Zealand. New Zealand has the advantage of being geographically located in a far distant part of the planet. So no wonder that, C. barnetti has not been reported there. There's some reports that in some countries like Norway and Iceland, some of those countries also have not seen a case of coxiellosis. Whether it's a matter of time, we do not know whether they will eventually find it there. Now, my focus is the Middle East, and almost every country in the Middle East has, in one or another, reported coxiellosis. Saudi Arabia and Oman have reported both in coxiellosis in humans and animals, and other countries like Jordan, Iran, Israel, and others have also reported the disease. Now, what about the UAE, the United Arab Emirates? There had been some reports of Coxiella positive animals, at least of serology, in camels and wildlife. Then, of course, there was no report in domestic animals until we came in and did this national serological survey in goats, sheep, and cattle, and were able to detect high rates of infection rates in these animals. The transmission, mainly, Coxiella barnetti is transmitted by inhalation of aerosols. It can also be transmitted by ingestion. And these aerosols come from animals that have given birth, the fetal fluids being aerosolized, or body secretions like urine, feces, and so forth, drying and being aerosolized. In fact, this is a risk factor for farmers, veterinarians, and other animals also which are in the proximity of this kind of environment. As you can see, urine, dry feces, and milk can be a source of infection. And of course, milk can be a source of infection for humans if it is not pasteurized. Then you also have indirect contact with fomites like contaminated clothing, instruments, and so forth that can be a source of infection for other animals and also for humans. Arthropods, many arthropods, ticks, for example, have been demonstrated to transmit Coxiella barnetti. Other insects also have been, tested, and many of them actually found positive on PCR, though the biological transmission has not been demonstrated in many of those. And what about in humans? In humans, you have transplacental or congenital transmission from mother to the child, blood transfusions, things like bone marrow transplants, intradermal inoculation, and there's also a hypothesis that this disease could be sexually transmitted. So this is a very nice schematic showing the transmission, and we shall also be looking at this in the epidemiology. As you can see, the human seated here and can get infected by inhaling aerosolized body fluids and so forth from animals, different animals. And within the animal population, you can have ticks maintaining that transmission cycle. So tick control would be one way of actually reducing the transmission rates in the population. Then, of course, you also have Coxiella in small animals, rodents, bats, which cannibals can predate on and also be infected through ingestion. And all these animals, including dogs, cattle, goats, sheep, can be a source of infection for humans. And unpasteurized milk is one of the things that can transmit the disease. So epidemiology, as I said, coxiellosis has a worldwide distribution. There is a probability that wherever every one of us is, there is this disease, with the exception of New Zealand. And of course, reservoirs. What are the reservoirs that maintain this disease in both in the wild and around, the human environment. Sheep, cattle, goats, dogs, cats. The first three are very, very important as reservoirs for human infection if you think of farmers and veterinarians who constantly interact with these animals. I've met a gentleman here in the biology department who actually isolated a Coxiella Barneti-like organism from birds. So birds can also maintain that cycle, reptiles and other forms of wildlife, even though, of course, there are arguments that these are not necessarily the same species as Coxiella Barneti. So they have called most of those as Coxiella Barneti-like organisms. Further with epidemiology, there is an occupational and environmental hazard. Livestock producers are predisposed to this disease. Veterinarians and laboratory technicians, vets, of course, who interact with these animals during treatment and other forms of disease management, and laboratory technicians who test the biological specimens collected from these animals. As I said, the first case of the disease was in abattoir workers who slaughter animals. Obviously, during the slaughter of animals, body fluids can be aerosolized, and these people can be infected through inhalation. Laboratory work, there have been cases of laboratory workers, at least in the US, where it has been documented that laboratory workers were infected by handling this organism. So let's briefly talk about fever in humans. As I said, it's usually acquired through the aerosol route or through aerosolized material, but it can be also, of course, acquired through ingestion. Now, of course, I'm repeating myself here. Aerosolization of fetal fluids, urine, fecal material, and that kind of thing can be ways humans are infected. And of course, the incubation period can be very long. Here, what I have cited here, 2 to 6 weeks, is even an underestimation. I think there have been some cases where they say it can go up to a year or if not more. And this Q fever, majority of the cases, 50% of those who acquire the organism are asymptomatic. But, a smaller percentage can get an acute disease. The acute form is characterized by this febrile flu-like disease with joint pains and headaches and so forth. And then the most devastating is the chronic form of the disease, which mainly involve the cardiovascular system, particularly causing endocarditis and specifically valvular endocarditis, which can cause damage to the heart valves, and that can require replacement of heart valves. That's very serious in terms of case fatality, of course. Of course, the acute Q fever is, as I said, is flu-like with all these symptoms, fever, joint pains, and so forth. And some of these cases can get atypical pneumonia, and that is characterized by a dry cough. And it can get more complicated when you get involvement of the liver in form of hepatitis. And so you will see, if you're examining such patients, you will see liver enzymes increasing. Skin rashes can be seen in some patients, and other signs like myocarditis, meningoencephalitis, and pericarditis can be seen. Death rate is, very low, but still of concern, 1 to 2% for the acute form of the disease. The chronic form of the disease is perhaps more devastating. 1 to 5% of the infected patients may progress to the chronic disease, including those who get the acute form of the disease. And as I said, this chronic form of the disease may involve the cardiovascular system, and patients who already have problems of heart disease are much more predisposed. Similarly, pregnant women are also predisposed to the chronic condition, as are the immunocompromised individuals, perhaps those have HIV infection or those who are undergoing chemotherapy for cancer and so forth. As I said, again, the chronic form of the disease is characterized by endocarditis, which can severely damage heart valves and cause cardiac insufficiency. And of course, that would increase the case fatality without intervention. Syndromes include things like chronic inflammation of the liver, which can progress into liver cirrhosis and liver failure, and then osteomyelitis, which is the severe inflammation of the bones, and osteoarthritis, which can have severe consequences. In the insert here, I lost my presentation. Did someone do something?
Dr. Barigye.
Okay. I just got my presentation back.
Oh, yeah, it's come. Come.
Yeah. So I'll quickly rush through. I was talking about the chronic form and risk to pregnant women. You know, it's very severe to get coxiellosis or Q fever in pregnant women because of the consequences. Of course, you can't get stillbirth, and if the baby survives, it will be very weak and there will be low birth weight. Placentitis is responsible for all these syndromes. And having said that, 98% of the pregnant women will be asymptomatic. Prognosis. Most of the cases are self-limiting. You know, this flu-like disease that we talked about in the acute form of the disease can be self-limiting. 2% of the individuals can develop severe disease and get active chronic disease. I think I won't talk much about chronic disease because I've just finished talking about it. And the case fatality rate will, be between 1 to 2.4 in case of the chronic form of the disease. Diagnosis, serology is very important. What we've been using here is ELISA, but the indirect fluorescent antibody test, microagglutination test, and CFT are very important. In our next project, we shall be using PCR as well. And isolation of the organism is very dangerous and not recommended because unless if you have biosafety level 3, and if you can avoid it because of the risks of infection. Treatment, tetracyclines are very effective as described in pregnant women, of course, and there you have to look at other alternatives. And patients who recover have long-lasting immunity. Now, of course, there are some vaccines which have been developed, not available in all countries, but they are there. So I hope that in the UAE here in the future, we can adapt such vaccines once we've finished collecting all the baseline data we need. Coxiellosis in animals, mainly in livestock species, sheep, cattle, and goats. The major demonstration or the manifestation of the disease is reproductive failure. Of course, clinically, the disease is asymptomatic, but you get abortion, stillbirth, infertility, retained placentas, and weak-born lambs and kids, and so forth. These recovered animals or animals that have aborted can be carriers in the herd, and therefore there's going to be continuous shedding of the virus. I have issues here. Okay. Other animal species, dogs, cats, horse, pigs are all susceptible. And as I said, with the first report of coxiellosis here in the wildlife ungulates, antelopes of different types, waterbuck, and so forth. Avian species are susceptible. And as I said, in all these animals, the disease is asymptomatic, except that, of course, it causes reproductive failure. Now, postmortem lesions for those who are pathologists, there is placentitis, and I would rather perhaps rush to the photograph that I have for you. There is placentitis characterized by a leathery thickened chorion and necrosis and purulent material, and with exudates on the cotyledons and within the intercotyledonary areas. So here's some pictures. Here's a picture of the placenta. These are cotyledons showing some, chronic inflammation. And I see, it's a specimen you may not easily appreciate. And if you see within the intercotyledonary areas, there are these areas of inflammation. And we have here a microscopic picture showing classic placentitis with perivascular inflammation and necrosis of the villi, chorionic villi. Here's a very nice picture that shows trophoblasts within the intercotyledonary areas. Coxiella barnett, in this case, of course, H&E, they're showing as basophilic organisms. The resolution is not very good, but we can see there are lots of organisms there. This is a very beautiful picture from one researcher, Dr. Agholm from Denmark, and I see a fluorescence in situ hybridization, and this is I see the fluorescence signifies there is a DNA for this organism. Diagnosis and treatment. Diagnosis, I talked about, serology, PCR, is very dangerous and very attempted, and treatment in animals. If you can't treat animals, say those have aborted from coxiellosis, you can treat them with tetracycline and reduce the rate of shedding the organism in subsequent years. Prevention. Good animal husbandry practices, for example, tick prevention, disposal of birth products by incineration or burying placentas and so forth can help. Separating new or sick animals and first testing them during the quarantine period to make sure they are negative. And the vaccination, as I said, is available for humans, and I know it's available in Australia, but not available in this country. On the human side, of course, pasteurizing milk is very important. Disinfecting the area with 10% bleach, particularly in the maternity ward. If you have, say, a dairy farm or sheep or goat farm, you can constantly disinfect the area where the animals give birth. Eradication is not at all practical because there are just way too many reservoirs, and there is definitely constant exposure, and the organism itself is very resistant. So this brings us to part 2. This relates to the work that I've done here. It's preliminary work, but I'm very happy to share it. When I arrived here, of course, as I said, I reviewed the literature and found there were cases, many cases of coxiellosis that were reported: Saudi Arabia, Jordan, Iran, Oman. And as I said, in particular with Oman and Saudi Arabia, they also Q fever in the people. And in the UAE, the only reports I could find were very few. After 1994 reported by serology of camel serum and actually had found positive cases in camels. Chava in 2012 reported it in Andres, in one zoo here in the UAE. And then, of course, recently we reported it in goats, sheep, and cattle. While we have reported a very high rates of infection rates, there is no scientific studies in the UAE to document whether there is zoonotic transmission. And based on the high rates of infection, on the basis of the serology data, I wonder if there is a coxiellosis problem in the region, and particularly in the UAE, because in some cases we found 60% was actually infected, rather seropositive. So not to repeat myself, I'll skip this data of, Afsar, who reported on racing camels, and Chaba, who reported on free-ranging wild ungulates, and talk about goats, sheep, and dairy cattle. We can see the average or the mean seropositivity rates: 32.15% for goats, 24.7% for sheep, 36.3% for dairy cattle, and most importantly, 41.4%, according to the data we have obtained on the dairy cattle with history of abortion, actually were positive. And there was a significant difference between those animals that had aborted and not aborted. So I think here we have shown some evidence that it is causing reproductive failure. In this table, as you will see, this is where we compiled data on showing the proportion of those animals that were seropositive and those that were seronegative. As I said, there was a significant, statistically significant difference between those animals which had, both those which were seropositive and negative. There was a difference, and later on, subsequent to that, animals that had history of abortion were significantly higher than those that didn't have history of abortion. Now you see that in this row, we're looking at different things, and I talk about Brucella abortus because when I got here, everyone was talking about brucellosis as being the cause of abortion in livestock, and the data doesn't show that. I understand there was brucellosis in the past, but I think there were some interventions, and currently we are not seeing that much in terms of brucellosis. So I wonder if those cases that were interpreted to be brucellosis, some of them, of course, are anecdotal reports, and I believe they could be coxiellosis. In here is a table, again, we are showing, this is where animals with the history of abortion. This is data from a dairy farm, and we can see that the animals with history of abortion, 61.9% were seropositive, compared to 32.7% of those that didn't have history of abortion. I already know the rest of the data, but you can see for yourselves how brucellosis or Brucella abortus compares with Coxiella barnett. Now, this is a graph from that data, which shows us the proportions of seropositive animals, those with history of abortion and those without. I say there was a significant difference between the two. And this paper has been accepted for publication in the Journal of Veterinary Medicine, and it's currently in publication. And here is a study in small ruminants, Again, I want us just to focus on goats and sheep, seropositive goats and sheep. There was definitely more goats, that are seropositive, and that has been consistent even in the literature from other workers. So again, this paper is also undergoing publication in the Journal of Tropical Animal Health and Production. So the research questions that are in my mind currently: is there a Coxiellosis problem in the region? There seems to be. Is there a Coxiellosis problem in the UAE? Is there ongoing zoonotic transmission in the country? If yes, can this One Health strategy help investigate this disease and contribute to the solutions for its control and management? So this brings me to the proposal that I've written, which is the basis of this presentation, as you can see here. Public and animal health significance of Q fever agent in the United Arab Emirates. Now, how does the One Health approach come in? I have mobilized a group from the College of Medicine and Public Health here at the same university where I am, and a group from my department. And we've had one meeting so far, and we have agreed to write this proposal. This is what we shall be doing in terms of research and getting the baseline data. The specific aims of this research will be to use molecular means, PCR and ELISA, to again appraise the prevalence of these diseases in livestock species, goats and sheep, all over the country. And we shall be specifically targeting, these Emirates: Abu Dhabi, Dubai, Sharjah, Ras Al Khaimah, and Fujairah. We shall also evaluate the association of this infection with abortion and goats and sheep, because we've demonstrated there is some evidence in cattle. Now we want to look specifically at small ruminants, and also want to evaluate the risk factors for infection in the livestock species. And this is the work which will be led by my team here, including myself as PI, and the work which will be led or which will take place at the medical school. We look at, test serum from farmers and animal workers to see if they are seropositive, and slaughterhouse workers to see if they are seropositive, and also evaluate the risk factors in those cases where human beings are found seropositive. And then we think, this is my references, we think that with this data, then we can start interfacing with the policymakers to inform them about the use of this type of bomb that I think is a very serious problem. I think I might have taken more of your time. Thank you very much, Dr. Maya, for inviting me, and I'll be very happy to answer some questions.
Now, the session is open for questions. Hello.
Hello.
Could you hear me?
Not very clearly.
Robert, could you hear me?
I don't hear you very well.
Yeah, could you hear me?
Now I can hear you.
Yeah.
No, I can't hear you
Dr.
now.
Robert, can you withdraw the sharing?
The what?
In the top, there is a stop sharing option. Yeah. Okay, thank you. Thank you.
Okay.
Okay.
So what was the question? Hello?
Hello, sir.
There was someone with a question.
I guess while that person is getting ready with a question, I might actually thank you for that wonderful presentation. We could see that, you have got experience across so many different countries in the world. And because, you were talking about the history of the organism, which was first discovered in Australia and USA, and the fact that you have been working across those two countries and now working in UAE where it's quite prevalent, I think that experience has been incredibly valuable for all of us. So the question is, why do you think it is so prevalent in UAE, particularly because there are countries where it is not at all prevalent, and you have showed us heaps of serological evidence to show that a substantial percentage of different kind of animals do have this organism, resident within them and are causing issues. So
Yeah.
why do you think is that prevalent? And what are the limiting factors that actually limits the organism to going to certain other countries? What do you think?
Yeah, I think, I'll start by comparing, for example, Australia and this country. The biosecurity system of, say, Australia and this country are not anywhere comparable. In terms of biosecurity, this country is a disaster. Of course, there is no clear animal testing. There are some government labs, but the work they do there, they mainly focus on the major transboundary diseases like foot and mouth disease and CBPP. And then the other thing is also the animal husbandry system here is very different. As you know, that's my hypothesis.
Okay.
Can you hear me?
Yeah, absolutely.
Yes, my hypothesis is the husbandry system in this country is like dairy farms. There is intensive animal husbandry where they have thousands, like up to 6,000 or even 10,000 of dairy cattle in one place, and they are all fed indoors and they are so close to each other. So if you think, and then they import animals, perhaps without screening them whether they are positive for Coxiellosis in this case, this sort of even applies to other diseases. I've been looking at leukosis. In zoonotic bovine leukosis is the same thing. Seroprevalence is very high. I believe that the animal husbandry system, animals close together, urinating, of course, with aerosolization of, feces and urine and that kind of thing might be contributing to a lot of this disease. And then there is also the traditional system. We do not know, we haven't looked at the traditional system where you have farmers living very closely to their animals. I would be So to summarize, I think the biosecurity system of this country needs to be strengthened. And number two, the animal husbandry system, there is a need for something to address there. And of course, we've given feedback to the farmers, but it's going to be very difficult, I can assure you.
Mm-hmm.
Yeah.
Yep. Absolutely. If anyone's, if anyone else is getting ready to ask another question, I've got just one more question before we go to the next person whilst someone else is getting ready.
Yeah.
So you did show us some of the serological evidence of its prevalence in domesticated animals. Mm-hmm. And I know that this is your hypothesis, but do you see zoonotic significance?
Yes, we haven't seen zoonotic evidence, at least it's not documented, but my hypothesis is there must be. One thing, like I said, biosecurity here is still very weak, but just across the border in Oman, they have reported human Q fever. Across the border in Saudi Arabia, they have. And we are seeing high seroprevalence in animals here. So that's why we need to get out and look at farmers or people who are constantly handling animals. In fact, we can include even our own veterinary students and see, if we can actually find seropositive cases. Now, when I went for a meeting at the medical school, they were telling me they have seen a number of cases with endocarditis. In fact, one of their hypotheses was they would be looking back retrospectively at those cases that have been treated for endocarditis to see if they were seropositive. If they can't get hold of serum from those patients, it would be very useful, to test and see if they're seropositive for Coxiellosis. And then, of course, we can also have prospective studies, hospital-based, now that we have a team at the hospital, and who will be looking out for febrile cases, people coming in with all these vague signs that could fit, chronic Coxiellosis.
That was quite an interesting perspective.
Dr. Neha is having one question. Dr. Neha, can you ask directly? Please unmute and ask.
So what are the potential causes of Coxiella burnetii as a biological warfare agent?
Actually, yeah, you're asking about that. The Americans are very worried that Coxiella burnetii could be suitable for use as a biological agent. I specifically left out a slide on that because I didn't want to go into the politics of that. because some of these things have been politicized here in the Middle East, as you know. So I didn't want to really tamper with that. But certainly anything that can be aerosolized can be used as a biological weapon. And we can see why they would use this. But there are certainly better candidates. In my mind, from my point of view, there are much worse pathogens that could be used as biological weapons rather than Coxiella burnetii.
Dr. Barigye, thank you very much for your presentation.
Yes.
You are doing a very good work there in UAE, and
thank you.
I see your research is need-based. You found a problem and you're trying to solve it.
Yeah.
Now, I want to ask you one question here.
Yes.
What is your predictions in terms of estimating the economic impact of Coxiella in UAE and actually in the Middle East at large?
Yeah. Yeah, Dr. Patrick, thanks for this question. I assume you're in Kampala, are you?
Yes, please. No, I'm in Jinja.
Okay, okay. So you know I'm originally from Uganda, though I've been around the world. Now,
oh, that's interesting.
Yeah, what I believe, of course, I mean, we looked at this dairy farm where they had serious problems of abortion, and they were worried about brucellosis. Of course, we tested these herds, and there was hardly any brucellosis. So, and they have had problems with abortion, and we have shown a statistically significant difference between those animals that had aborted and those that didn't have a history of abortion. So it's definitely having an economic impact on dairy herds. We haven't looked specifically at small ruminants, but certainly, you know, anything that's going to cause reproductive failure is going to have some economic impact. I have a colleague here who has specialized in the economics or agribusiness. He's actually a specialist in agribusiness. And this is one thing that we could be studying in the future. Now, we would need, first of all, to build the data. This is just the baby steps we are making. Once we have solid data, then we can attract funding to address some of those issues. But it's so amazing to see that 60% of the animals being positive is very scary. It's very scary.
Are there some of the interventions, maybe some kind of
now you will be surprised. You will be surprised. On the books in the UAE, they have Q fever as a notifiable disease. But there had never been any documentation other than 2 serological studies, one in 1994 and the other one in 2012, of the disease. Now, I don't know what they will do. Once we have solid data and we take it to them, they have to come up with interventions. They have to. Some of the dairy farmers, we went and talked to them. It's very complicated here because dairy farms are mainly owned by the sheikhs, who are the rulers. And, you know, it's very difficult to reach them and pass on this information and actually explain it to them so that they can appreciate the gravity of the problem. Just like the traditional farmers back in East Africa, they love to see thousands of heads of cattle, and they are not going to even destroy the positive animals. You can't do that. I don't think they will welcome it. But we shall have to give this information to the policy makers in the relevant ministry, and then they will have to come up with interventions. And I would be very happy at that time if they invited me to give my opinion.
Yeah. Yeah, that's interesting.
Yeah, very interesting.
Thank you very much.
Thanks, and I hope to meet you when I come to Uganda.
Sure, no problem.
Yeah. Sure.
Yeah.
Any more questions?
So, Dr. Robert?
Yes, sir, I have one question, sir.
Yes, please. Yes, please.
Thank you, sir, for the informative presentation. Sir, my question is, sir, what about the seroprevalence of Coxiellosis in canines globally?
I didn't hear the question.
Seroprevalence of Coxiellosis in canines.
In
canines, dogs.
Canines. So we haven't looked at canines. Someone needs to mute their mic. Yeah, we haven't looked at dogs. But certainly we need to. Canines can predate on small rodents and stuff like that, or even can eat uncooked meat, which is positive. And I think in this kind of environment, not only dogs are being exposed. I always wonder, even as humans who consume these products, obviously the milk, if it's pasteurized, we are safe. But we should be looking. Let's start with the livestock species and build the database to include canines and other animal species. In fact, a species of interest would be the cat. Those who don't know about the UAE, there is a very huge population of feral cats on the streets. And definitely those who would interact with small animals, small rodents. There have been, I think, Coxiella has also been isolated in small creatures like cockroaches and so forth. So I think the cats might play some role in the epidemiology of this.
Thank
you, Dr. Barigye, for excellent presentation. I have one simple doubt. Actually, I had my research work for PhD on Coxiellosis only, and we targeted ruminants, mainly cattle, and we found very good seroprevalence, 10 to 20%. But the problem is the association of Coxiellosis with abortion cases, because in India we are having many other pathogens like brucella is very, very common and very endemic in many parts of the country, and we can have leptospira. So it's really difficult to rule out other pathogen. Means we can't say that the Coxiella is associated with abortion. So actually I faced this problem in proving during submission of my research papers. So just I want to know how you used to rule out these confounding pathogens.
Yes. That's a very brilliant question. If you remember the tables I showed, there were other pathogens we were looking at, and all can cause abortion. In fact, in sheep and goats, we looked at Chlamydiophila abortus, brucella, toxoplasma, and that kind of thing. The reason we included those were to remove the confounding factors. For example, when I arrived here, everyone was talking about brucellosis. I said, well, we need to look at all the others. Now, something we haven't looked at is Campylobacter, for example. Also the viruses that we could possibly look at. And it's because of the limitations of the budget in my first project that I didn't look at those. I had a plan to look at the whole range of those organisms. Now, with my background as a diagnostic pathologist, I did a lot of abortion investigations in the US. And so one of the strategies I intended to use in the upcoming work is actually to perform necropsy examinations on aborted fetuses. I didn't go through the details of how we intend to implement that, but I'll be collecting aborted fetuses and I'll be screening them for all sorts of abortigenic agents. So if we do that, we use different tests, of course, we use molecular tools, we use immunohistochemistry and many other tests. We can build a case for Coxiellosis. Now, of course, also the mathematicians come in. We have one guy is a brilliant statistician who's been analyzing this data. He's an associate professor of statistics here. So far, he's looked at the serology data and analyzed it in the best way possible to create a data association, to demonstrate the association between abortion and seropositivity. But certainly, we are not satisfied until we do molecular studies for all those range of organisms that could cause abortion.
Okay, thank you so much. Thank you so much for your.
That was a good question.
Okay, thank you so much.
Yeah, that was a brilliant question.